Make sure it's correct. Okay. Okay. So let's have a class. Now, everybody should have your results. Let's talk about those tubes first. Okay. So you have those 3 tubes, fermentation. I just want somebody to show others the results. We have glucose, lactose, and sucrose. Okay, let's look at the E. coli. Who has E. coli? Whose inoculation is E. coli? Okay, you guys. So I'll just bring one as an example. Is that okay? Okay, when you see here, so everybody look at the glucose. What do you can see? For glucose, it's completely yellow. Can you see that? Which means it's acid fermentation positive. And when you see on the top of the Durand tube, there's a bubble, which means it's gas positive. I just give you an example. I'm not going to go over everything. So for example, E. coli. You can look at your own stuff, okay? E. coli, you will see. It's gas positive and lactose positive. This is showing you yellow color, and the gas you will see the Durand tube has bubble. Okay, very clearly. Now you can see, let's say sucrose. This is also E. coli, you didn't see anything. That means a little bit of pink, but definitely no gas. Now let's look at lactose. You see the same thing. See lactose? It's yellow color, and on the top there's a bubble. So for E. coli, you will see lactose. This is glucose fermentation also positive. And the lactose fermentation positive, it's yellow and there's a gas. So I just give you an example, a typical example of E. coli. You can look at your own to record it. Because I don't know. I don't know every bacteria, how they grow, but that's a typical example for E. coli. And it's a good example for 3-tube fermentation. Okay, so that's the first one. Second one, let's look at your urea tube. Who has Proteus vulgarius? Who has Proteus vulgarius? Give one tube to me. Just one. For Proteus vulgarius. No, yeah. The urea tube. I want the urea tube. Yeah, look at the urea tube. Okay? So we have Proteus vulgarius, and this is E. coli. And Lee, you can take a picture of this one, if possible. This is the one very obviously is one positive, one negative. Okay, I want to just, so you can see these 2 tubes. Don't take me, just take a picture like this. I can put on the eCampus possible. So urea tube, you will see. We have Proteus vulgarius, which is, you see it's pink, or we say it's cherry red. That is positive. And the E. coli, which is actually is, or we say just the original color. So just the original color. That's why it's negative. Okay. I believe all of the sample, all of the bacteria you have, Proteus vulgarius is the only one is positive. Most of the others are negative. So I'll return this back to you. Okay. Now you can record your situation on your bacteria. I mean, I don't know everything. Like I said, Staphylococcus aureus might be also negative. But I know Proteus vulgarius is positive, so I just gave you an example. Proteus vulgarius is positive. Okay? This is the first thing. Second, this is second. Third, let's do catalase test. Okay, you should have agar. I said you have Staphylococcus aureus and E. coli. What we should add there? We should add H2O2. Is that right? I want everybody to do it right now. Add H2O2 on you. Find the agar place, which has Staphylococcus aureus and E. coli, which is you do for your catalase test. Then you add H2O2 there. Do it. Don't look at me. Just do it. Yeah, just add H2O to. Just give to all there. Yeah. Very easy. Just add. You will see one side is bubble. Very obvious. Just add it there. On two sides, add some. Just dump some. You see one side is bubble. Very obvious. It's like a boring. Which side? Is that Staphylococcus aureus? Yeah. So this side will be very obvious that you see a bubbling. And E. coli is none. So Staphylococcus aureus is catalase test positive. And remember I said that is to differentiate Staphylococcus and Streptococcus. Okay? I had a video which is on the YouTube channel already. So did everybody find it? Looks like boring, is that right? Bubble comes out. That's very important. But remember, you should never do this on blood agar. Otherwise, you pour it, you find it. Because blood agar has oxygen there. Okay? Number 4, I want you guys to test called oxidase test. So oxidase test, let's do that. Karen, can you do like this? We have only 2, so I want to save a little bit. Just 2 of them share one place, you put 1 drop.
Okay.
And Lee, if possible, you can do the other side. Just to put one drop there. Let's use that one. 2 of you can share one, and just 1 drop. Just one drop. So you're gonna have the place ready, which is Pseudomonas and E. coli. So Pseudomonas aeruginosa, which is PA side, and E. coli. We just gonna add a couple of drops there. 2 of you share one place. You can share with your batch partner. Okay, then you can see which side is. Just 1 drop each side. Very quick, just one drop each side. You see which one turns purple. Then you'll see which one is positive. It's very obvious within 1 minute. You have to do this within 1 minute, remember. Which one is positive? You should see one side, very obvious is purple. You guys can share one, yeah, okay? Is that good? Which one is purple? Is that Pseudomonas? Is that Pseudomonas that is purple? Okay. So Pseudomonas is oxidase test positive and E. coli is negative. Now this is very important test. This is a test used to compare Pseudomonas aeruginosa versus a group of bacteria called Enterobacteria ACA. We will do the ACA test real quick. The name called Enterotube, because this is difficult to differentiate them. The reason is all these bacteria are gram-negative, large shape, facultative, lactose fermentation positive, gas fermentation positive. So it's very difficult to differentiate it. That's why we use oxidase test. Can do a very easy identify clinically, okay? Okay, that's good. Now number 5. Let's look at your 3 plates. You have your 3 plates. We talk about the bacterial enzymatic function. Let's look at you. First of all, look at your skim milk agar. We have Bacillus cereus and E. coli. Which side you find a transparent zone? Skim milk. Is that Bacillus side? Look at your plates. Don't look at me. Is that Bacillus side you see a transparent zone? Good. Okay, so it's positive. E. coli is negative. Okay, the second one, look at your split blue agar. Which side is transparent zone? We have Staphylococcus aureus and the E. coli. Is that Staphylococcus aureus part? Should be. Which means Staphylococcus aureus has lipase. This guy has lipase. And this guy, Bacillus, has caseins. Which means protease. Okay, now a little bit complicated for the last one. This is Starch Agar. What you should do? You're not going to see the transparent zone, is that right? What you should do? You're going to pour the iodine there. We see which side is turned brownish, is that right? Okay, I want to... Who has that? Who has the plates? Can borrow one to me? Which is a Starch Agar. Okay, thank you. So you have a Starch Agar. We put Bacillus and E. coli. So Lee, we wanted to do an iodine conformation test. So what we're going to do? We're going to add iodine. So I'll do it. You can do it yourself. I want a gram iodine. Where has the gram iodine? Some of the iodine is being used a lot, so I will find one iodine.
This one right here.
Okay, just use one to me. I can do this for you, and you can come here to take a picture. So we want to be putting these on the side. Okay. We want to go to the other side. We're also going to put it. We want to mix it. Now, I want to borrow, who has another one? Who can borrow me another one? Can I have your plates? Can I have your starch plates? I hope it's good. Okay, I'll do the second one, Lee. So we're going to be doing this. We pour the iodine here. We pour the iodine here. We mix it. You should be seeing the starch which is reacted with these, and one side will turn brownish color and the other side is not. So it seems like from here it's not very obvious. I didn't see which side will turn brownish color, the other side is not. Now, not very obvious. I don't know, maybe it's old. So this one I can return back to you. Back to you. You can take a look. Which side turns brownish, which side is not. You actually can pretty much see that. That turns brownish, this is like steel black. So you can see that. So which side is shows brownish? E. coli side, is that right? E. coli side you can see a brownish color. And this one, you see the iodine is on the top, but did not really turn brownish. So what that means? You see the brownish, which means starch is still there. So starch present. And on the other side, you didn't see brownish, which means starch absent. Absence. So which means this Bacillus subtilis is amylase test positive. Okay. Remember, I said that we need to twist it a little bit of your mind. Okay. When we see there, you see the starch side is actually E. coli side, there is a brownish color, which means the starch is present. So the amylase did not use it, which means they don't have amylase. But on the bacillus side, you see starch is absence, which means the amylase is there because they hydrolyze the starch. Let me see one more time to see what it looks like. But it's been there a little bit of a while. Now the idea what you should do is that this side you should leave a little bit space. Then we'll see the difference. Here we probably pretty much we cannot see anything. Okay, but that's okay. So that's also the results what we talk about. Okay. So we talk about all these results. You're gonna have to record it. Typically, that first couple ones. Okay, we're gonna still continue to do 3 tests today, including 1 enter tube test. Let me see how many we have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. We have 13. Maybe everybody can use one. I have 25 for 1 box. I assume in the second lab will be 8. We'll see. We maybe just do a pair, okay? For the enter tube test. So today you should receive 2 fact sheets. One is for biochemistry testing bacteria 2. Another one is called enter tube test. And the API 20E. API 20E, I'm not going to do it here. I wrote it there. You can read it through. It's a multi-channel test, but it's more expensive and more complicated. Enter tube 2, I have it. It's like a pencil. So we will do it. Looks like we're going to do a pair, which means 2 of you do 1. Because this one box is about $300. This one probably one channel might be like 20 bucks or something. So I just want to save a little bit. Okay. Now other tests we will do 3 more tests. So what are those 3 tests? Number 1 is called the SIM test. SIM test we call it 1 shot, 3 jobs. Okay, so what are they? S means sulfide. I indicates indo. And M means motility. Now, this test is a little bit of difference between what we did before. We are using loop the whole semester so far. But this test we will using is needle. Okay, so this test is a little bit different. You need to have a needle ready. Then you have this tube. So you have a needle go inside straight like this. So Lee, do you want to take a picture for me? So you take it out. You will be put this needle in, straight in, and then go out. So that's 1 shot to do 3 jobs. Don't use loop. I'll tell you why. Okay. So that's the first thing you need to know. 1 shot, 3 jobs. Use needle, not loop. So first of all, why we have to use needle? We're gonna first talk about the motility. How do we know bacteria having the motility? This agar contains 0.5% melted agar. So this is a semi-solid medium. Now, how do we know a bacteria have real motility? How do we know bacteria do not have real moving motility? We put needle in. We want to see, right in the middle, we want to see where the bacteria are going to move in after incubation. So after the incubation, if the bacteria stay in the middle, which means bacteria do not have motility, which means no motility. In other words, if you see bacteria moving around, you see bacteria everywhere, cover the whole. That means it has strong motility. Now, how do we know bacteria have a strong motility? This determined by flagella. Okay, do you still remember what is called monotrichos? What is this called? Amphitrichos. What is this called? Lithotrichos. What is this called? Petrotrichos. Is that right? Flagella, which means the bacteria has a real motility. Now, this is for bacteria. What is for fungi? One, two, three, four, five, six, seven, 8, 9. Fungi is 9 plus 2. Flagella becomes flagellae. This do the spin. Okay, that's a real movement. So just let you know. We using semi-solid agar. Use needle injection to see the bacteria whether they have motility or not. Depends where are they after incubation. If right in the middle, no motility. Goes everywhere, they have a strong motility. It depends what kind of the flagella they have. That's why we're gonna find it. Okay? This is the first thing. The second thing what we want to talk about is sulfide. Now here we're going to talk a little bit about anaerobic respiration. So you need to know why we do sulfide test. Some of the bacteria, their electrophile acceptor is not oxygen. It can be other inorganic chemical. For example, sulfide. Okay? Then the bacteria could be using sulfide and reduced to become SO4 sulfide acids. Okay, they can be going through anaerobic respiration. They can use the energy, but the energy, of course, going to be lower than aerobic respiration because of this difference between the voltage value of the initial donor and the final acceptor is smaller than aerobic respiration. Now, how do we know the sulfide has been reduced? Become sulfide gases. Inside of this agar, there is an ingredients. There is called metallic ingredients. For example, iron is inside. Once the bacteria has been reduced to sulfide, the iron will turn black. So if you see 2 weeks from today, so we have a holiday next Thursday, next Tuesday, 2 weeks from today, if you see your tube is black, which means it's sulfide positive. Because the metallic ingredients like iron inside of this medium will turn black. Now we left one, which is called indole. What is indole? This is byproducts of tryptophan, TRP. So some of the bacteria can use amino acid like a tryptophan. Now what is tryptophan? This is tryptophan. This is TRP tryptophan, okay? Some of the bacteria can use tryptophan. They break down right here. Now what are the two parts? This side is indole. How about this side? This is amino pyruvic acids. Now why it is important? Every time when you see this guy, pyruvic acids or pyruvate, they could go either way. We call it a metabolic intermediate. They could go fermentation, they could go phosphate, or they could go aerobic respiration TCA cycle. So once you see these kind of intermediate metabolic products comes out, it could be let bacteria survive, go any metabolic pathway they like. So we're gonna testing whether bacteria use the tryptophan. So we testing whether bacteria generate indole. How we test it? We're gonna have this tube, okay? You use needle injection. Already goes in, goes out. How do we know it's positive? 2 weeks from today, you're gonna add one drop of a large drop of a reagent called Kovac. If this Kovac turns cherry red, which means it's indole positive. Okay? So the work is very easy. You just put a bacteria there, but the story behind that is more important. So that's called SIM test. 1 shot, 3 drops. That's the first thing what we're going to do. Okay, second thing, we call it phenylalanine test. Or we easier to say PHE test. That is another example for some of the bacteria use amino acids. So what is phenylalanine? Not this one. Phenylalanine is this is phenylalanine. Now, what is the phenylalanine means? It's amino acids. Bacteria can use it. Now, how bacteria use it? They do deaminase. What means deaminase? Which means they take out amino acids, base. They take out of this guy. Okay. Once they take out of this guy, what they become? What is this part? Phenylpyruvic acids. Now, what is phenylpyruvic acids? What is this guy right here? Is that glyceroldehyde 2 phosphates? Glyceroldehyde 3 phosphates? Then phenylpyruvic acids, then pyruvate. What is this called? Glycolysis, is that right? That's why this guy is important, because they can